We are looking at question number 8 from GATE Biotechnology 2017 question paper. So the question is, if the nucleotide composition, that is the percentage of a viral genome, is A is equal to 10%, U is equal to 20, C is equal to 40 and G is equal to 30, which one of the following is this genome? So option A, double-stranded RNA, option B, single-stranded RNA, option C, single-stranded DNA, and option D, double-stranded DNA. Before we answer the question, let's brush up some facts and terminologies about viruses. So viruses are infectious particles that are composed of nucleic acids and proteins. They are obligate intracellular parasites, which means that they can't be considered alive or dead. So they're just sequences, that is like nucleic acid sequences. But when inside a host, like a human, so when inside a host cell, they come to life, they replicate, and they cause infections. So viruses are very small in size, ranging from 20 to 300 nanometers, and can be seen only with an electron microscope. So tobacco mosaic virus was the first discovered virus, which is a plant virus. And some of the basic terminologies are shown here. So virion is a complete virus particle. So you can see that this is a virus particle. It's made of a nucleic acid and it has a capsid, which is a protein, and both of these together account for the naked virus, and this is covered by an envelope which is also composed of proteins. So this whole infectious particle is called a virion. And prions are infectious proteinaceous particles, that is the protein component of the virus that contribute to the infections are called prions. And then bacteriophages are viruses that infect the bacteria. And virome is the general collection of all the bacterial and eukaryotic viruses, and virology is the branch of science that deals with the study of viruses. So the viruses differ in size, shape, and symmetry. So there are three major types of capsid symmetry, and one is icosahedral or cubic. So it has 20 faces, like an each, which is an equilateral triangle. And one of the examples of a virus that has this kind of a structure is adenovirus. The other configuration is helical structure. So the protein binds around the DNA or the RNA, whatever the genetic material, the nucleic acid sequence is. So the protein binds around that in a helical fashion. So one example of such structure is the tobacco mosaic virus. And the other structure is a complex structure which is neither cubic nor helical. So examples for these are bacteriophages. Now speaking about the viral genomes, which is the nucleic acid component of the virus, the genome may either be DNA or RNA, and this could be either single-stranded or double-stranded. So the genome may also be either positive sense or negative sense. That is, positive sense RNA is equal to the mRNA, so it can be immediately converted into the protein. Whereas the negative sense RNA is complementary to an mRNA, and first they need to be converted to the positive sense RNA by RNA polymerase before translation to proteins. So this is how they infect the host cell. So positive sense viruses, they can directly cause the infection by getting themselves converted to the protein, and sense RNA, they have to undergo an additional step. So they have to synthesize the complementary RNA sequence first, and then they replicate and they translate. There are certain exceptions for these as well, and some examples are retroviruses. So they make DNA from the RNA using the enzyme RNA dependent DNA polymerase or the reverse transcriptase RT enzyme. So examples of these retroviruses are the human immunodeficiency virus, which causes AIDS, and avian myeloblastosis virus, which causes diseases in birds. And the other examples is hepadnaviruses or pararetroviruses. These are DNA viruses that carry reverse transcriptase, and the DNA genome is transcribed into RNA, which acts as template to make new viral DNA strands. So this reverse transcriptase enzyme generally has three enzymatic activities. So the first one is RNA dependent DNA polymerase. This synthesizes a DNA strand which is complementary to the RNA template. And then the RNase H removes the RNA strand from the DNA RNA hybrid double helix. And then there's DNA-dependent DNA polymerase, which completes double-stranded DNA synthesis. So unlike other DNA polymerases, this reverse transcriptase enzyme lacks a proofreading capability and therefore has high error rates during DNA synthesis, like up to 1 error in 2000 bases. So this high error rate of viral reverse transcriptases provides selective advantage for the survival of the virus in the host system. So this also makes it very difficult to design or synthesize vaccine against a virus, because they replicate very fast, and the sequences can evolve and change very quick. So that makes it hard for vaccine development. So again, here's an outline of the viral DNA genome configurations. So they could be either DNA or RNA and can be linear or circular as well. And also they could be single-stranded and double-stranded, like you can see here. And some of the examples for each of these categories are shown here. For example, tobacco mosaic virus is an RNA virus. It has a single-stranded linear genome. Whereas the hepatitis D virus, again, is an RNA virus. It has a circular single-stranded RNA genome. So the viral genomes can also be either segmented, which is composed of multiple pieces of nucleic acid, or non-segmented. So the genome segment is a unique piece of nucleic acid among the multiple pieces of the genome. For example, influenza A virus has a segmented genome comprising of 8 single-stranded RNA segments. And herpes virus, they don't have segmented genomes, but they are comprised of one linear double-stranded DNA molecule, and have the so-called unique long and unique short segments. And HIV, the human immunodeficiency viral genome, carries 2 copies of the same single-stranded RNA molecules. Hence, the genome is considered non-segmented, but they are just called copies and not segments. Now, based on their configuration of nucleic acid, like whether they are DNA or RNA or sense or antisense, or whether they carry a reverse transcriptase enzyme with them, all the viruses are categorized into 7 different classes based on the Baltimore system of classification of viruses. For example, the class 1 viruses are double-stranded DNA viruses, and one such example is a herpes virus. And for example, class 4 viruses are positive stranded, like sense single-stranded RNA viruses. And examples for these are polio viruses, and so on. Now, coming back to the question, so we are given with the composition of the viral genome. So the nucleic acid bases that are given are A, U, C, and G. And so first we need to find out if it's DNA or RNA, and then we can find out whether it's single or double-stranded. So based on these nucleic acid, the bases, we could see that we have A, U, C, and G, and there's no T. So we know that RNA, they do not have thymine, and instead they have uracil. So from this example, it's clear that the virus is an RNA virus and not a DNA virus. So the options C and D are ruled out because it is clear that the viral genome is composed of RNA and not DNA. Now, to know if it is single-stranded or double-stranded, we could see that all of these proportions add up to 100. So 10 plus 20 is 30, 30 plus 40 is 70, and 70 plus 30 is 100. So, and we know that in a double-stranded structure, adenine base pairs with uracil, A base pairs with U, and C base pairs with G. So in case of a double-stranded structure, if there's 10% of A in the genome, the proportion of U should also be 10% because they base pair with each other. And C, the proportion of C should be equal to G. But in this case, that is not what we see. So clearly the virus has a single-stranded RNA genome, which answers our question. So the answer to this question is option B. So if the nucleic acid composition of a viral genome is as follows, which one of the following is the genome? So this given virus is a single-stranded RNA virus.